Okay, so that was coronary circulation. That great cardiac vein is going to run in parallel with the anterior interventricular artery. That small cardiac vein runs in parallel with that right marginal artery. And that middle cardiac vein ran in parallel with the posterior interventricular artery. And of course, the added feature of the coronary sinus for the venous circulation. So one main goal of our heart anatomy is to trace blood flow through the heart, pretend that you are a red blood cell, and move through all of these vessels and valves and chambers and see where you end up as an entire circuit. So pulmonary circuit, systemic circuit down here. So here's your label for that. This is going to be oriented to your worksheet for the day as well. We already kind of did it. We're going to kind of do this backwards and forwards. I even have a video from a previous semester of me acting this out. I'm not going to do it on this recording. I'll act out with the class and do this. So systemic veins to the superior and inferior vena cava, to the right atrium, through that right AV valve, to the right ventricle, through the pulmonary semilunar valve, to the pulmonary trunk and arteries, gas exchange in the lungs, back to the pulmonary veins, which take you to the left atrium. Now we're on the left side of the heart, through the left AV valve, to the left ventricle, to the aortic semilunar valve, to the aorta, to the systemic arteries, and then finally, gas and nutrient exchange in peripheral tissues is going to take us back to that right atrium. So I'm going to finish up this lecture, and then once I've stopped recording, we're going to revisit that concept. We're going to turn the entire classroom into a heart, and we're going to assign you each a chamber as groups, and you're going to tell me if I'm deoxygenated or oxygenated, where I came from, where I'm going, what valve I'm taking to get there, etc. So you're going to have to be the heart. And it seems to be very effective when we actually do that. Now, continuing with the lecture for now, back to the histology. Recall that, again, we have this branched pattern of the cells. We have intercalated discs with gap junctions. That there's a lot of folds at those intercalated discs. What's the purpose of folding? Surface area. It's always about that surface area. So more communication between cells. So as long as it's not electrically insulated, that electrical impulse is going to be shared by every cardiomyocyte all at the same time. Cardiac muscle has a lot of similarities with skeletal muscle in the sense that we're going to have sarcomeres that are our contractile units. We have the same striations with the M line along the middle, the Z discs at the end of each sarcomere. We're going to have the same actin and myosin filaments. We're going to have the same power stroke. We're even going to have, for the most part, the same ions being the basis for this contraction. So just something to think about for the future, that sodium, that potassium, that calcium, that was the same thing that was responsible for skeletal muscle contraction, that was the same thing responsible for neurons firing, is also going to be responsible for cardiomyocytes contracting. Sodium being a really important ion for depolarization of that membrane. Okay, I think that's about it. One important thing about cardiac muscles is that they have a long absolute refractory period. So remember, with neurons firing onto muscle cells, you have a refractory period in which that neuron cannot fire again. Same thing with muscle cells. You have a period in which that muscle cell cannot depolarize again. In the heart, it's actually fairly significant that this be lengthy. There are going to be a variety of heart conditions related to premature heartbeats. We want to avoid premature heartbeats. We want to keep that heart rate relatively steady, and we want those electrical signals to go through and not be able to occur right away again. Yes, what's up? Yes, absolutely. Her question was, would AFib be one of those? And the answer is absolutely. So if we didn't have a long refractory period, we could simply contract our heart muscles and it would stay contracted. That's the definition of a tetanic contraction. It is not the same as tetanus, which is a bacterium that causes tetanic contraction of extensor muscles. That's why tetanus is called tetanus, and tetany is chronic contraction of a muscle. So same word root, different concepts. All right, last big concept for today is the conducting system of the heart. Your heart is kind of magical. Your heart has modified cardiomyocytes that initiate their own action potentials. So it's not exactly neural tissue that's initiating the heartbeat, but it's going to be cardiomyocytes that act like neural tissue, that send actual action potentials, just like your neural tissue does, which is really, really cool. This is called autorhythmicity. Even without any autonomic innervation, your heart is going to create its own heartbeats. In fact, the autonomic innervation to the heart is only going to speed up or slow down the heartbeats that are already happening. So what do we think that parasympathetic nerve is going to be innervating the heart? Vagus is correct. It's always vagus, right? It's always going to be vagus. Until we get to the pelvic cavity, it's going to be vagus. So that vagus nerve is actually going to innervate this pacemaker of the heart rate here, and this one as well. But it's actually just going to slow down your heart rate. In the absence of vagal innervation, your heartbeat would be 100 beats per minute, approximately. So the other way to say that is this SA node, the sinoatrial node, initiates action potentials at 100 beats per minute. I'm going to give this to you the long form, complicated, scientific way, and then I'm going to give you my mnemonic device that's going to make it so easy that you'll feel really dumb. Okay? So, SA node initiates action potentials that sends electrical impulses via the internodal pathway between nodes, internodal pathway. That sends signals to the AV node, the atrioventricular node. That AV node is going to send signals along the atrioventricular bundle. There's an old school term, bundle of His. That's going to get through that fibrous skeleton. So recall that we've got to have insulation between atria and ventricles, but they also have to communicate. So this is the pathway by which the atria do communicate with the ventricles. So there's a delay between the SA and AV node, so that this contracts first, then this contracts.
Is the node just the circular part?
So the circular part is the node, correct? And then there's the AV node. So SA, internodal, AV, AV bundle. Then that bundle branches. Literally, it branches off into a left and right bundle branch. And when that reaches the apex of the heart and starts to come back up the walls of the heart, we call those Purkinje fibers. So here is my super dumb, super effective way to remember that. Save his bundles, Purkinje. That's it. SA to AV, the bundle of His, Purkinje fibers. That's it. So that's what this looks like. SA node depolarizes the atria. Internodal pathway takes it to the AV node, which delays it for just a bit so that it can send it to the ventricles but not have them contract at the same time. That AV node propagates down the bundle of His, the AV bundle of His, branches into left and right bundle branches. At the apex, they become Purkinje fibers. And that whole structure is going to cause depolarization of the ventricles, which causes contraction of the ventricles. So that green that is illustrated throughout this, that is the spreading of the impulse, causing depolarization of those action potentials within the cardiomyocytes, which is going to correspond with muscular contraction.
the thing that's sending is the action potential?
So the SA node is initiating action potentials, and it is propagating through these pathways. Yes. The action potentials, yes. SA, AV, bundle of his, bundle branches, Purkinje. Yes.
So, it's in your slides. I was just curious if you're going to go over how many beats at each.
Yeah, they do have that specifically. I don't care about specific numbers. You should probably remember 100 beats per minute. What's our definition of 100 beats per minute? Tachycardia. So that may indicate, for example, not exclusively, but for example, either sympathetic activation or problems with your vagus nerve. So that's why you might want to remember 100 beats per minute there. Generally, that number just goes down. So by the time you get to the AV node, it's only propagating at 50 beats per minute. By the time you get to the Purkinje fibers, you're beating at 30 beats per minute. And again, the significance there is that this system can get blocked. We have something called a right AV block. That is a thing that happens. If that gets blocked, then the ventricles may slow down compared to the atria. So when we start talking about arrhythmias, that's when this comes into play. It's not completely arbitrary that we're making you learn the electrical conducting system. There's a reason for it. Okay, one more concept before I stop talking for the day. Just very brief ECG, just so that we can have fun with it with our ECG machine that I've pretty much just stolen from the lab. We're not going to do anything with numbers, not really. We're going to learn what these major waveforms are. It goes like this: P, QRS, T. Yep. I will. I'm trying to speak to this right now, but you have a P wave, then you have a QRS complex, and then you have a T wave. And that's not the scale that I've drawn on the board. The P wave indicates depolarization of the atria, which means that atria are contracting. So that's the electrical signal that means atria are about to contract. The QRS complex, that's the Q, that's the R, that's the S. It's much larger. That's because it's depolarization of the ventricles. And even if you go back and look at this electrical conducting system, you can think of it as a QRS if you want to, or QRS. So again, this is depolarization, which corresponds to contraction of the ventricles. The T wave is a measurement and the change across the ventricles. Again, this is repolarization of the ventricles. So the T wave is typically slightly larger than the P wave. QRS is obviously the biggest change. What this is literally a measure of is the overall change in charge across the entire heart. This is a summation of many, many different action potentials happening in many, many different locations in sequence. And when we shove all of those changes in voltage together onto one image, then we get this typical ECG pattern. So people who work in depth on ECGs are going to know a lot more about the individual waveforms than is being portrayed here. Our Netter's coloring book actually describes this very, very effectively. So I can distribute this to you guys later if you would like to understand that a little bit better. So just because it's repolarizing and depolarizing, it's still a change in charge, so it still is going to register in this positive direction. So really all you need to know to start having fun with the ECG is atria contract, ventricles relax. And when you've gotten there, then we can start to appreciate a normal ECG pattern. We can look for it on ourselves with our ECG machine. This does enable you with these numbers to count how many beats per minute are occurring. Again, just kind of broad strokes here. Every bold blue line is 1/5 of a second. So if you have the same peak repeating every 5 of these, that means 1 beat per second. So if this one, there's a QRS, there's an R right here, and then 1, 2, 3, 4, 5, there's another R. 60 beats per minute is correct. So that's all you need to do for today to have fun with it. We could go into more detail, but I think I'm happy with my recording for now, so I'll stop lecturing.